Okay, I'm Ken Houck. I get to introduce Don Hilvert. But before that, I should just say Bill and I go back a long way. Congratulations, Bill, to this for this great honor. Bill and I were both at Harvard 50 years ago. Hate to say those words, but we didn't really know each other then. But ever since I've been at UCLA, Bill. I know. I'm much older than Bill. I have to admit that, yeah. I have even less hair than Bill, I think. So anyway, but to get serious, Bill has been a fantastic faculty member throughout the time I've been here. Has really been a superstar of our faculty, not only in science, which we hear alluded to throughout today, but in his service to the department, he's really given his all for our department and for UCLA, so thank you, Bill, for that. So I'm going to now introduce Don Hilvert, who's also a good friend of mine. He's professor of organic chemistry in the ETH Zurich, and he's a world leader in creating new proteins with designed catalytic properties. Don has advanced our understanding of molecular origins of how natural and designed enzymes achieve their awesome catalytic power and selectivity. And he does other things too, as you're going to hear today. Don graduated from Brown University, another Ivy League school, and then he went on to Columbia and worked with the great physical organic chemist Ron Breslow. Unfortunately, we lost Ron Breslow a month ago, but the great contributions he made to physical organic chemistry live on in a lot of physical organic chemists, but especially in Don and in our own Eve Rubin. Don was then a postdoctoral fellow with Tom Kaiser at Rockefeller University, and he joined the Scripps Research Institute as an assistant professor in 1986. And we at UCLA tried to lure him away in 1996, but instead he moved to ETH Zurich as full professor of organic chemistry in 1997. Don's won a number of awards. He was a Sloan Fellow, an Arthur C. Cope Scholar, a recipient of the Pfizer Award in enzyme chemistry. He was a recipient of the Kaiser Award 2009 of the Protein Society, and he received an honorary degree from University of Uppsala in 2011. He was elected to the American Academy of Arts and Sciences recently, 2016. So Don, as I mentioned, is a physical organic chemist who's made important early discoveries about catalytic antibodies. And then he has been involved in enzyme design with David Baker, and we've been honored to be part of that as well. And Don has used directed evolution to achieve world record activities of designed enzymes after he carries out directed evolution on these primitive designs. Don's an expert in selenocysteine incorporation at enzymes, semi-synthetic enzymes of many types, and capsid engineering that you're going to hear about today. He's talking about design and evolution of supercharged protein containers. Don.
Okay, so thank you very much, Ken, for that very generous introduction. I'd like to say my thanks to Bill for the privilege and honor of being here at the celebration of his award of this year's Glenn Seaborg Medal. I met Bill exactly 7 years ago, almost to the day, at a birthday party for Arya Warshel held at USC. And we sat next to each other at lunchtime, and he told me about his work on virology, a field that I knew woefully little about, basically nothing. And based on those conversations, I'd like to tell you about what they inspired in my lab in Zurich over the last several years. So specifically, we've been interested in designing new cages that utilize electrostatic interactions to bind various cargo molecules. So you're all familiar with viruses. We've just heard about viruses. They package their RNA and DNA genomes in these closed shell proteins and deliver them to cells. But they're not the only protein containers that are around. Ferritin, for example, is a very important protein that mineralizes iron nanoparticles. Proteins can be folded in cage-like structures, and protein microcompartments, these bacterial structures that are the equivalent of eukaryotic organelles, are something that Todd Yates has studied here at UCLA. They co-localize enzymes and promote mini-metabolic pathways. The carboxysome, which is responsible for fixing most of the CO2 biologically, is one example of this kind of organization. These natural protein compartments have been expanded recently, also largely due to Todd Yates' efforts in collaboration with David Baker to use cubic point symmetries and computational design to create a huge number of completely unique structures that you could also imagine exploiting for binding various cargo molecules. Now, the benefits of encapsulation are manifold. Perhaps most simply, by placing something inside a container, you link genotype and phenotype in a very simple way. You can also protect the contents against degradation. You can sequester toxic materials from the environment. If you're looking at coordinated enzymatic activities in one of these confined environments, you can concentrate substrates with the enzyme so that you get very efficient conversion. You can control the fate of reactive intermediates. You can minimize side reactions. And for applications, you can imagine using these vessels to deliver molecules to cells for imaging, and ultimately to create artificial organelles that provide a metabolic advantage to their host organism. So in my lab, we have focused on a natural bacterial microcompartment called lumazine synthase. Lumazine synthase is a protein that self-assembles into a 60 subunit closed shell particle. Here is a cutaway view of this. It's about 16 nanometers across. Unfortunately, you're not going to be able to see any of the references, but I can tell you later on if you'd like. This has been studied by Adalbert Bacher in Munich and by Ladenstein in Sweden. Ladenstein actually determined the structure. Lumazine synthase is interesting because it itself is an enzyme. It catalyzes the conversion of a diaminouracil into this compound called lumazine. And that happens on the inside of the cage. That's an odd place to place an active site of an enzyme, except that those organisms that form these closed shell structures also encapsulate the next enzyme in the pathway leading to the vitamin riboflavin. So riboflavin synthase sits inside this capsid, and it takes two molecules of lumazine to create riboflavin and regenerate the diaminouracil. So this is a very efficient way of taking a molecule that's generated inside this luminal space and sending it to the active site of the adjacent enzyme without releasing it into the environment where it can get lost or precipitate. Now, we recently discovered that the encapsulation of riboflavin synthase is mediated by a 12 amino acid peptide attached to the C terminus of the enzyme. It's very hydrophobic and presumably interacts with sites on the capsid interior. We know that the encapsulation occurs as the capsid is being built up biosynthetically. Once you form this particle, it doesn't come apart unless you denature it. So you need very, very strong conditions to disassemble this structure. Well, before we knew that lumazine synthase used these peptides to direct association, we wondered whether we could take this capsid and turn it into a more generalizable encapsidation system using charges. So our simple idea was perhaps we can just paint the interior of the protein shell with negatively charged amino acids, and then perhaps it would be able to bind complementarily charged positive cargo molecules.
What's the native charge distribution inside?
It's roughly neutral. So this is the monomer. The cage looks a lot like T1 equals 1 virus particles, for those of you who are familiar with this nomenclature. But the monomer has no structural similarity to natural viruses. It's basically a flavodoxin-like fold with a core sheet packed by helices. The N and C terminus project to the outside, and we have these loops that project into the interior. And by design, Florian Seebeck and my group basically introduced just 4 glutamates on the surface, which would increase the interior charge by 240 for the native 60 subunit protein. That's interesting because that means we can potentially encapsulate positively charged molecules such as HIV protease. And that's interesting because producing HIV protease in bacterial cells is difficult. It's a protease, so it chews on other proteins and is quite toxic. And if you encode this, or if you place the gene encoding the enzyme behind a tetracycline promoter, you can regulate the amount of enzyme that's produced in the cytosol. And what we find is that even at very, very low concentrations of the inducer tetracycline, we don't see any growth whatsoever. However, if you place the protease in a cage, for example, our negatively charged lumazine synthase, you place a physical barrier between it and its targets in the cell, and now you have growth. You basically protect the host from the ravages of this toxic material. And that means you can do evolution. So you can carry out random mutagenesis and then multiple rounds of selection for variants of the enzyme, of the capsid-forming enzyme, that encapsulate the HIV protease more efficiently. And we did this for 4 rounds, and what we found is that we obtained variants that had 7 new mutations that enhanced our original charge-based encapsulation mechanism. So what we see is the introduction of 3 new negatively charged residues. We see the disappearance of 2 positively charged arginines and their replacement by neutral residues, and then 2 neutral changes. So now we have a really negatively supercharged molecule. And that leads to the preferential encapsulation of our target enzyme. So here you can see a Western blot. We're detecting the HIV protease produced in the cells using an antibody. And if we have no capsid, you can see that when we induce the gene with 700 nanograms of tetracycline, you don't see any HIV protease whatsoever. If you use the wild-type lumazine synthase, same story. But with Florian's negatively charged capsid, we now see a very distinct band. And after evolution, this was carried out by a graduate student, Binja Rösdorfer, you see a 10-fold increase in the amount of this toxic protease that's produced within the cell. What's interesting is, in contrast to the wild-type capsid, you can isolate the empty lumazine synthase variants. This is from Aquifex aeolicus, a thermostable organism. So the capsids are also highly thermoresistant. You can isolate these without any guest, and then you can add guests in vitro and see spontaneous encapsidation. So this can be a 5 nanometer gold particle, it can be a 15 nanometer gold particle, it can be a supercharged capsid. This is a supercharged version of ferritin that we engineered. It has a large number of positive charges on its surface, and you can see this has a diameter of about 12 nanometers, and we can put 3 or 4 of these within the interior of our capsid. And we just mix the 2 together. We wait some period of time, isolate the capsids, and it's encapsulated. And since the ferritin still will nucleate the mineralization of iron particles, we can actually localize these iron nanoparticles within this confined space. In fact, this is quite general. If you have a supercharged, positively supercharged protein, it will go spontaneously into the capsid. In fact, we've used this supercharged version of GFP, because it's fluorescent and very easy to monitor, that David Liu at Harvard had engineered some years ago. It has 36 positive charges on its surface. Basically, you mix the 2 together, you wait a little bit, and it will go inside the capsid. But this can also be very useful as a tag. We can fuse the GFP to an enzyme or any protein that we're interested in, and it will spontaneously go into the capsid structure to make a small nanoreactor. In fact, we can put 2 different enzymes by making 2 fusion constructs so that we could imagine starting to do a mini metabolic sequence within the confines of this cage particle. And so we've used this quite successfully for a number of applications. So for example, we've encapsulated a peroxidase and used it to catalyze the polymerization of a diaminobenzidine. So you can see that we can make spatially confined polymers that depend on the size of the capsid that we use. We've been able to sort substrates. Here we've encapsulated a protease, the TEV protease, and we've been able to change its intrinsic substrate specificity for negatively charged substrate molecules, which is seen here. So the negatively charged substrate is cleaved preferentially over the positive substrate in the absence of capsid, but as soon as you place the enzyme in the interior of this negatively charged particle, then we preferentially cleave positively charged substrates, as you see very nicely in this case. So we can completely change the intrinsic substrate preferences of the enzyme by localizing it in a specific microenvironment. We've also made primitive versions, primordial carboxysomes, in which we've co-localized carbonic anhydrase and rubisco and have looked at the kinetic effects of co-localization. How does this happen? I mean, it's kind of a surprise that you can just mix these two proteins and then spontaneously form a cage complex. Now, what I haven't told you is that when we did the design, there was actually a very significant structural transformation. So the wild-type cages, as I told you, are closed shell and have a diameter of about 16 nanometers. When Florian put all of the negatively charged glutamates on the interior, we nearly doubled the size of the capsid. Now we have a structure that's about 29 nanometers in diameter, and when Binya evolved this, it increased even further now to 36 nanometers. So what's going on? Many viral particles are also polymorphic, and this is very well understood. You can go from T equals 1 to T equals 3 to T equals 7 type structures. Is that what's going on here? Well, we didn't really know, and in order to address that question, you have to know something about the stoichiometry of these cage structures. Now, this has been characterized, and so we teamed up with Michael Heck at the University of Utrecht, who is a mass spectroscopist who is developed native mass spec methods that allow him to determine the masses of these huge, huge complexes. And so he took the wild-type lumazine synthase and was able to see a beautiful charge envelope for the intact particle. And when you deconvolute this, you see that it is a 1 megadalton structure, exactly as you would expect for a 60 subunit capsid. So we then took the lumazine synthesis, the ALS-NEG variant, the expanded version, did exactly the same kind of experiments, sees again a beautiful charge-resolved envelope of charged states. And when you deconvolute this, what you find is that you have a 3 megadalton structure, which would correspond exactly to what you would expect for a 180 subunit molecule. So this is very similar to a normal T equals 3 state, which typically contains 12 pentamers and 20 hexameric building blocks. For the more highly evolved variant, he wasn't able to get nice charge resolution, but based on the peak maximum, we estimate that this is at least a 240 mer and perhaps a 420 mer. Now, we tried very hard to crystallize ALS-NEG and ALS-13, the optimized variant, but we weren't very successful. And I met our next speaker, Jack Johnson, at a physical virology meeting in Leiden 3 years ago, and he presented this absolutely fantastic data by cryo-EM on a virus, showing that you could even see the individual side chains. So I went back to Zurich and I told my coworkers that we really have to explore this technology. So you're all aware of the enormous advances in cryo-EM. This is evident from the Nobel Prize of this year. This is based on fantastically high-resolution direct electron detectors and fantastic software. And I'm not going to go through the details here for lack of time, but we decided to try this in collaboration with Nenad Ban, my colleague at the ETH, to look at these structures. So we initially started with the wild type because its crystal structure had been determined by Ladenstein many, many years ago. And Eita Sasaki, postdoc who had 0 structural biology experience, was able to do this experiment. So he took the cryo-EM pictures of the initial particles, was able to sort them, do 2D classification, then assuming icosahedral symmetry, was able to obtain an initial model. He could then do the 3D classification and then ultimately get interpretable density, which is shown here. So this is a structure at 3.9 angstrom that was based on 3,000 particles. Now we know the structure of the individual pentamers, and we can actually fit them into the density very, very well. And what we see is that the wild type is, as we expected, a dodecahedron consisting of 12 of these pentameric sites. And you can see here the little hole in the middle of each pentamer through which the diamino uracil has to pass. So that was a success, and we went ahead and printed this. So what about the design? Here we don't know what the structure is, but we do exactly the same routine. We obtain the original particles, do the 2D classification. Here, when we tried to fit a model for an icosahedrally symmetric particle, we failed to get interpretable density, so we lowered the symmetry to tetrahedral. We got a solution that gave us good 3D classifications, and then ETA was able to solve the structure. So this is at almost 6 angstrom resolution based on an analysis of 26,700 particles. And again, you can see the pentamer fits in. And you also notice that we have 3 symmetrically distinct pentameric units. These are shown in blue and cyan. These form trimers of pentamers, and they're joined together by these dimers of red pentamers. So completely unprecedented structure with tetrahedral symmetry. And we printed this guy as well. So it's a little bit bigger than the original. And then we moved on. So what's striking here is that the 180 subunits are assembled as 36 pentamers. There's no hexamers whatsoever, as you would see in a normal icosahedrally symmetric T equals 3 like virus capsid. So what about the most evolved variant? So we did exactly the same routine. This time we could get icosahedrally symmetric solution. So during the optimization, we moved away from icosahedral symmetry and then returned to it in the most advanced version. I just turned something off. And then here is our 3D classification. And so here is the large structure. This is about 5 angstrom resolution, 9,000 particles. And you can see that, again, it forms almost a dodecahedral shape. And you can see one of the pentamers surrounded by 5 other pentamers makes one of the dodecahedral faces of this molecule. So this is 360 subunits. Again, it consists only of pentamers, in this case 72 pentamers, no hexamers again. Again, an unprecedented structure.
Excuse me, can you remind us which variant of the wild type this is? We saw the negative one where you put...
So this is after evolution. So it has more negative charges. So it's just gotten bigger. So we start with the wild type capsid, an interior volume of 270 cubic nanometers. We go to the ALS neg, and we increase the interior volume to about 4,000 cubic nanometers. And then our largest one is almost 60 times bigger than where we started. So this is the ALS 13. Now, what happened is, in retrospect, quite understandable. You introduce a large number of negative charges on the interior luminal surface of this capsid. What are they going to do? Repel one another. And that's going to lead to a change in the cone angle of this pentameric building block for the capsids. So this will become more and more cylindrical. And as a consequence, when it packs against another pentamer, the angle that they distend will decrease. So we go from pentamer-pentamer interaction of 63 degrees in the wild type to 34 in the ALS neg to 20 in the most evolved variant. Now, if you take ALS wild type and the most evolved variant, which both have the same icosahedral symmetry, you can also see how the individual building blocks interact with one another. So this has 60 identical pentameric units. In this case, we have far more. And we have 2 symmetrically distinct variants. We have the central pink pentamer at the 12 vertices, and the icosahedral faces are made up of this trimer of pentamers. Now, Todd Yates told us that this kind of patterning is quite reminiscent of the tiling that Kepler looked at 400 years ago. And you can see as you try and tile a larger and larger surface, you can't make a closed cell structure. Inevitably, you introduce these defects. And these defects are pores into the interior of the capsid. In the case of ALS-neg, we have 12 of them. In the case of the ALS-13, we have 30 very large pores. In fact, in the interior here, the diameter is about 4 nanometers. And that's exactly the size you need in order for a GFP to pass through the pore into the negatively charged interior, essentially without barrier. And in fact, we know from this paper that the binding of the supercharged GFP to the interior of the cage is essentially diffusion limited. It's controlled by ionic strength, but it occurs at the diffusion limited rates. So we can add our capsids then to this collection of computationally designed protein cages that Todd and David have introduced. What's different here is that we have introduced mutations very distant from the subunit interfaces, whereas these are based on creating sterically and chemically complementary faces of the interacting building blocks of the pentamers, of the capsimers. And in this case, the changes are farther away and introduce more subtle geometrical alterations that lead to these large structures. There are many questions that these results raise. Because of the nature of the interaction of the cargo molecules and the capsids, the density of packing, particularly in the largest capsids, is not very high. This is in contrast to the natural microcompartments like the carboxysome that Todd studies. Can we create much more dense packing? This is a challenge for us. Can we control the assembly and disassembly of these structures? Can we gate the entry and exit of the cargo molecules? That would be useful, for example, for delivery applications, if we could release them on a particular external cue, change in pH, et cetera. Can we generate other structures from these same building blocks? And then in the context of today's symposium on virus structures, can we actually bind negatively charged guests? So in principle, we could start again. We could take our original lumazine synthase and paint the interior not with negative charges, but with positive charges, and hope that these structures will then bind negatively charged molecules, for example, RNA. In this case, the switch of computers moved the negative charges. Sorry about that. So Ken Wojciechowski, a former postdoc of mine, has actually done this with a wild-type lumazine synthase cage and was able to observe that these could encapsulate short RNA sequences when produced in bacteria, E. coli. But they're certainly not able to bind anything as long as the gene encoding the capsid molecule. If we would like to move towards nucleocapsids that resemble virus-like particles, that's going to be absolutely essential. And so we decided to pursue another strategy. So many RNA-binding viruses have long extensions that project into the lumen that are positively charged. They often will contain a lot of arginines. And that's because many of the natural viral proteins have the N and C termini directed to the interior of the capsid structure. Well, that's not the case here. And so we need to re-engineer our monomer, and we decided to do that by circular permutation. We're going to link the N and C terminus, cleave the polypeptide chain between two residues at the bottom, and then we're going to add an arm, an arginine-rich motif. And so here is what we designed. So we took a 12 amino acid linker to connect the original N and C termini. We introduced a short linker, and then we have this arginine-rich motif. So these still assemble into capsids. They're not the wild-type size. They've been expanded. They are about 30 nanometers in diameter, so similar to the ALS-neg structure, the middle structure that was circulating. And the specific arm that we chose is from a termination peptide in lambda phage called lambda N+. This binds a very specific RNA sequence, the hairpin that you see here, and it makes particular interactions with the loop nucleotides. This is called the box B sequence. So we added the box B sequence to the 5' and 3' untranslated regions of the gene encoding our capsid, hoping that we would be able to isolate capsids that could encapsulate their own gene. So we translate this into the building blocks, and if everything works, this should package its own message. Well, this works to an extent. We're able to generate capsids that bind RNA, but the largest RNA is about 200 nucleotides. It's not even probably 200 nucleotides. Far, far shorter than the 863 nucleotides of the gene segment. And so to optimize these structures, we turn to directed evolution. Probably one of the problems here is that intracellular RNases are able to cleave the message itself before it gets encapsulated. And if this structure is a lot like ALS neg, it's going to have big pores through which RNase can go through and cleave up anything that's bound. So we need to evolve this to package more quickly and to prevent this internalization of the enzyme. And to do that, we're going to use a strategy very similar to the packaging that you saw for the positively charged guests. We're going to take our capsid. I've removed the box B sequences, so we can translate this, but we're going to produce it in the presence of a gene encoding the HIV protease that's now equipped with the box B segments. And hopefully we're going to get packaging. And if we are successful, that will prevent translation of this toxic gene product. And that would give us growth. And hopefully the RNase will be prevented from entering the capsid and digesting the message. So we actually did multiple rounds of evolution. We isolated variants of the capsid. Then we took them and cloned them into a vector in which they now again have their box B sequences. We're going to transform E. coli cells with these constructs. We're going to translate the message. We're going to produce the protein. And hopefully, if everything works, we're going to encapsulate the gene encoding the molecule that surrounds its own gene. We then isolate the capsids, and to put pressure on them to make sure that they are only able to load full-length RNA, we are going to treat them with benzonase, an engineered 60 kilodalton nuclease that cleaves DNA and RNA. So anything that's hanging on the outside should be completely digested. We're then going to extract the RNA, and then we're going to do reverse transcriptase. And then we repeat this process 3 times, and then we remutagenize. And then in the last step, we do 3 more cycles with a much smaller RNase A, which is about 120 amino acids, and can easily pass through the pores of the wild-type ALS molecule to look for variants that can protect their cargo from the nuclease. And this shows you the results of the experiment. So this is the runoff transcript of the message of the circularly permuted RNA. And the original lambda CP, the circularly permuted variant, is able to encapsulate RNA, but as you can see, most of the RNAs are very, very short. Here would be 200 nucleotides. So you may see some faint bands up here, but most of the RNA that's bound is very, very short. And after the first couple of cycles in which we treat the capsids with benzonase, we're able to select for variants that are able to encapsulate full-length capsid. And then after an initial introduction of mutations and screening, we can amplify the amount of RNA that's encapsulated by a factor of 4, and we increase the amount of full-length RNA very substantially, as you see in this gel. If you do quantitative reverse transcriptase PCR, what you can see that about 10% of the capsids have full-length gene. The intermediate is much less. In the wild type, you don't see any detectable levels of the full-length gene. So what happened in this process? So here is our starting circularly permuted variant. In the first round of mutagenesis and screening, we introduced 6 mutations that are distributed really all over the protein. This has not changed the size, but you still see some heterogeneity in these molecules. And after the treatment with RNase and another round of mutagenesis and screening, we introduce 5 more mutations, and we get these very regular capsid structures. So we've analyzed the contents of the capsids using RNA deep sequencing. And what we find is that for the most evolved variant, most of the RNA is derived from the gene. It's not necessarily full length. Remember, only 10% of the capsids have the full length gene. But the rest of the fragments turn out to be enriched in fragments of that original gene. So the protection is not complete. We see a little bit of RNA from the plasmid and a little bit from the genome. This is dictated by the box B sequences that determine the selectivity of the interaction. If we remove those, we can still encapsulate RNA. This is presumably just because the gene encoding the capsid is produced at very high levels using a T7 promoter. So mass action will necessarily lead to some encapsulation. But only a much smaller fraction of that RNA is derived from the gene. Much more comes from the plasmid and from the genome. So the basic message is that it's pretty easy and it's pretty fast to take a non-viral capsid protein and convert it into something that is able to create a nuclear capsid much like a virus particle. So this supports some of the hypotheses regarding the origins of viral capsid proteins from natural proteins that happen to be in the cell. Now again, there are many open questions. Probably the most important here is what is the structure of the evolved alpha 9 variant? We don't know yet. It's the same size as ALS-neg, but presumably the pores have been reshaped or closed, perhaps by the lambda N plus peptide, because we see significant protection. But we don't yet understand the mechanism in detail. Can we further optimize loading so that we can get a much higher fraction of capsids with their own genome? Can we package larger RNAs? In the gel that you saw with the alpha 9, there are actually some of the RNAs that are packaged are twice as long as what we started with. So that opens up a lot of possibilities of generating more complex functions in which we encapsulate messages that have perhaps 2 gene products on them. So in the end, the use of supercharged structures is very useful for packaging a large range of cargo molecules based on very simple electrostatic interactions. And we're very excited about exploring the possibilities of using these, for example, to produce toxic proteins in a cell, proteins that would be otherwise very difficult to produce in a recombinant fashion. We can use them to control the storage. We can store and release the guest molecules in controlled fashion. Can we create nanoreactors that actually provide an advantage to the host towards the construction of artificial organelles? And so that's the story that I'd like to leave you with this afternoon. This has been a fantastic group of co-workers who have made all of this possible. This was started by Florian Seebeck. The original evolution was carried out by Binya Versdorfer and Ken Wojciechowski. Eita Sasaki was the person who solved all of the structures of the original designs. And Naohiro Terasaka, with some help from Yusuke Atsuma, have been able to create these new structures that are able to bind nucleic acids. So I'm very thankful to them and to our collaborators and to the sources of funding. Finally, many congratulations to Bill on this year's Seaborg Medal. Thank you.